Good laboratory practices, GLP, are a set of regulatory guidelines and standards designed to ensure the quality, reliability, and integrity of non-clinical laboratory studies. Definition: a quality system related with the organizational process and the conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived and reported. GLP is a formal regulations created by US FDA on November 19, 1976 and designed as new part of 21 CFR Guidelines, Part 58 in 1979. In 1981 OECD, Organization for Economic Cooperation and Development, produced GLP principles that are international standards. GLP principles: 1. Quality Assurance Program. 2. Testing Facility Organization and Personnel. 3. Storage and retention of records and materials. 4. Reporting of study results. 5. Performance of study. 6. Facilities. 7. Standard operating procedures. 8. Test and reference substance. 9. Test system. 10. Apparatus, materials, reagents. Schedule L1, Rules 74, 78, 150E. Schedule L1 is set of regulatory guidelines under the Drug and Cosmetic Act in India. Schedule L1 specifies standards for the quality control, testing, and documentation needed to ensure that pharmaceutical products meet safety and efficacy standards. Key aspects of Schedule L1: 1. General requirements. A. The laboratory or organization must be legally authorized and accountable. B. Management must ensure the lab follows good laboratory practices, GLP. C. Qualified quality manager must oversee all technical activities and implement quality system, reporting directly to top management. D. Quality manager also schedules and oversees GLP audits by external experts appointed by top management, and maintain quality system as per quality manual. 2. Premises. A1. Laboratories must be designed to prevent insect and rodent entry, as well as cross-contamination. A2. Interior surfaces should be smooth, crack-free, and easy to clean and disinfect. A3. Adequate space and utilities, water, power, gas must be provided for testing and equipment. A4. The air ventilation system must ensure a dust-free environment. B. Laboratories should have sufficient lighting, ventilation, and, if needed, air conditioning, to maintain appropriate temperature and humidity, for testing and equipment functionality. C. The drainage system should prevent water logging and facilitate maintenance. D. Tabletops must be smooth, crevice-free, and resistant to acids, alkalis, and solvents. A. Biomedical waste must be destroyed according to the Biomedical Waste Management and Handling Rules, 1996. F. There must be adequate space for storing reference and working standards, with SOPs for their maintenance and evaluation. G. Air circulation must be maintained in areas where sterility tests are conducted as per Schedule M. H. Bioburden control should be managed in both controlled and uncontrolled areas, example, airlocks. 3. Personnel. A. Laboratory staff must have the necessary qualifications, proper training, and adequate experience for their duties. B. A training record for all personnel must be maintained. C. The head of the laboratory must have high professional standing, experience in drug analysis, and laboratory management. Laboratory head responsible for C.1. Ensuring document control and maintenance, including raw data, SOPs, documentation exhibits, protocols, and training charts. C.2. Planning and organizing quality system audits and managing corrective actions. C.3. Investigating technical complaints. C.4. Taking final responsibility for recommending regulatory actions in cases of non-compliance. 4. Equipment. A. The laboratory will have all necessary equipment for its activities. B. Analytical instruments must be kept in a dust-free environment. Temperature and humidity conditions should be maintained and monitored regularly. C. Instruments, benches, and surroundings must be clean and tidy. D. Calibration of instruments must occur at regular intervals with records kept. A. Equipment records should include equipment name, manufacturer's name, model number and type, serial number, date received, current location, condition on receipt, instructions, calibration and maintenance frequencies, calibration records. Responsible staff, authorized users, history of damage or repair, list of spares and accessories. F. A progress register for non-functional equipment should be maintained. GSOP for preventive maintenance should be prepared. Equipment like burettes and pipettes must be checked for calibration accuracy. I. Maintenance procedures should be outlined in SOPs, and servicing should be done by specialists. J. Defective equipment should be labeled out of order and calibrated after repair. K. Maintenance for services like electricity and gas should be handled by competent personnel. L. Autoclaves must meet operational, safety, and validation requirements, with records kept. M. Work involving harmful vapors should be done in a fume cupboard. The fume cupboard's exhaust system and water drainage must be checked frequently to ensure proper function and avoid waterlogging. Store chemical and reagents. A. Store and handle chemicals safely, considering their properties and hazards. B. All reagents and solutions must be clearly labeled. C. Maintain a register with raw data and SOP for preparing and standardizing stock and volumetric solutions. D. Stock and standard solution containers must include name of the chemist who prepared it, date of preparation, use by date based on stability, standardization records. A. Transfer hazardous chemicals using proper equipment. Avoid makeshift or unsafe methods. 6. Good housekeeping and safety. 8. General and specific written safety instructions will be distributed to all staff and revised periodically through posters, audiovisual materials, and seminars. B. SOP for safety, housekeeping, and loss prevention will comply with Government of India regulations and include B.1. Safety data sheets must be available to staff prior to testing. B.2. No eating, drinking, or smoking in laboratories. Human food must not be stored in working areas. B.3. Staff must wear lab coats, gloves, masks, and eye protection as required. B.4. Adequate first aid kits and firefighting must be accessible. Staff must be trained in their use. B.5. Operators must wear sterilized garments, including headgear and face masks. B.6. Staff must be educated in first aid, emergency care, and antidotes. B.7. Safety rules for handling compressed gas cylinders must be followed. Staff should know color identification codes. C. Laboratory precautions. C.1. Water showers must be installed in appropriate locations. C.2. Use rubber suction bulbs on manual pipettes. C.3. Provide warnings and instructions for dangerous reactions. C.4. Facilities for waste collection, storage, and disposal must be available. C.5 and 6. Staff must be trained in the safe disposal, corrosive materials, and handling of potassium cyanide and cyanogen bromide. C.7 and SOP for chemical and biological waste management will be established. 7. Maintenance, calibration and validation of equipments. All laboratory equipment and instruments must follow proper methods for testing and calibration, and they should be regularly calibrated and validated. B. Calibration frequency may vary by instrument, taking into account the manufacturer's recommendations, staff experience, and daily usage. C. Each laboratory should prepare SOP for calibration and maintain a logbook for documenting calibration results. 8. Reference material. A. Reference materials are vital for testing and calibration. They must be traceable to authorized agencies. B. Laboratories prepare working standards by comparing them with reference standards. Purity is checked regularly. C. New reference materials should have a code number assigned and documented. Working standards also need an identification code. D. Maintain a register with details like source code number, receipt date, batch number, assay values, storage conditions, and expiry dates. A. Check working standards before use to ensure they haven't deteriorated. Record all observations. Store materials as recommended, keeping those at 2 to 8 degrees Celsius in a refrigerator and avoiding freezing if advised. 9. Microbiological cultures. A. Laboratories must prepare a SOP for maintaining and subculturing microbial cultures. B. Non-viable or mutant cultures must be destroyed by autoclaving, conducted by authorized personnel. Limit passages to no more than 5. C. All activities must occur in an aseptic area by authorized personnel. D. Standard biochemical tests on subcultures should be performed to ensure viability, as outlined in the literature. 10. Quality system. A. Measurements and calibrations must conform to compendial requirements. Validated methods must be followed. B. The system must ensure compliance with planned arrangements for activities, processes, techniques, or practices. C. It should help detect and correct non-conformities early.
D. Remedial actions must be taken based on internal and external audit observations. A documented quality policy must be in place for the organization.
11. Internal quality system audits. A. Internal audits ensure the integrity of analyses. Audits are conducted periodically with a set schedule and procedure. An appropriate checklist verifies compliance quality system and regulatory requirements. Only trained and qualified personnel conduct audits. Auditors must be independent of the activity being audited. B. The head of the laboratory sets the audit schedule. Each activity must be audited at least once a year. C. The head of the laboratory is responsible for corrective actions. D. The quality manager handles non-compliance issues. The quality manager analyzes problems and takes necessary actions. A. The quality manager maintains records of analyses and audit protocols. 12. Management review. Top management must review the quality system at least once a year, covering the following: 1. Internal audit reports. 2. Issues from previous reviews. 3. External audit reports, if any. 4. Surveillance reports, if any. 5. Proficiency testing results. 6. Complaints, feedback from users. 7. In-house quality control details. 8. Need for system or documentation updates. 9. Induction training for new staff. 10. Any other laboratory requirements. 13. Standard operating procedures. Characteristics. 1. Written in chronological order for lab activities including analysis or calibration. Point 2. Must include SOP manuals and periodic review. 3. User-friendly documents and specify that the person responsible for each task. B. Key areas for SOPs. 1. Sample handling and accountability. 2. Receipt, identification, storage, and sampling. 3. Record keeping, storage, retrieval and reporting. 4. Computerized data systems. Point 5. Audit operations, inspections, and reviews. 6. Maintenance, cleaning, calibration of instruments. 7. Equipment failure handling. 8. Analytical data methods. 9. Raw data. 10. Data handling and storage retrieval. 11. Health and safety protection. 12. Change rooms. 13. Entry-exit. 14. Storage and maintenance of microbial culture. 15. Maintenance of sterility room. 16. Use and storage of reference standards. 17. Procurement of stores and equipment. 18. Monitoring of testing of samples. 19. Method of retention of unexpended samples, their location, maintenance and disposal. 20. Document control. 21. Redressal of technical complaints. 22. Housekeeping. 23. CAPA. 24. Working procedure, test method. 25. Calibration manual. 26. Training manual. 14. Protocols and specifications archive. Laboratories must maintain a specification archive with current versions of necessary specifications, shall be kept as per IP. B. All updates and corrections noted in master volume of Pharmacopoeia to prevent obsolete sections, supplement and addendum also available in the laboratory. C. Specification archive shall contain the 1. List of all pharmacopoeias. 2. File on test methods for patent and proprietary medicines. Validated test methods that meet compendial parameters like precision, accuracy, reproducibility, specificity, linearity, and ruggedness etc. These methods must be submitted to the Drug Control Authority. A. Raw data refers to all types of laboratory records, including handwritten notes, photographs, software, printouts, and data from automated equipment. It also includes records of environmental monitoring, equipment calibration. B. Changes to data must be made by striking through the old entry, noting the correction, providing a reason, and signing with the date. For automated systems, the responsible person must be identified at the time of data output. C. Data integrity and security must be ensured, with access restricted to authorized personnel only. 16. Storage and archival. Residual samples must be stored for 1 year after the final report. B. Laboratories must have procedures for managing quality documents, collection, indexing, retrieval, etc. C. Raw data, documentation, SOP, protocols, and reports must be archived for easy retrieval and protected from damage or loss. D. Storage conditions must ensure security and confidentiality of original documents. A. Paper documents should not be stored in high humidity, and raw data on tapes or disks must be carefully preserved. F. Optical disks must have a lifespan longer than the storage period. G. Thermal paper data should have a photocopy retained to prevent fading. H. Document retention times must be defined. Reference Guidelines 1, Drugs and Cosmetics Rules 1945 Schedule L1, 2, 21 CFR Guidelines Part 58, 3, ICHS 7A2.11, 4, WHO Handbook of GLP and PIC Guidelines 5, OECD Principles of GLP.